People change. Opinions change. Places change. Thoughts change. Time never stands still. At Surrey, we're adapting and striving for better. Teaching advances, learning develops. Our approach evolves, methods move on, progress. Student needs evolve. We listen and respond. Connections deepen. Support grows. Facilities improve. Global partners connect. Relationships grow. New faces arrive. Lives are changed. Research evolves. Knowledge broadens. Collaboration inspires us. Discoveries are made. Communities are brought together, taking care of each other, reaching out, learning together. A pandemic ensues. The world stands still. We join forces, take steps forward. The nation applauds our heroes. Surrey heroes. We're in this together, growing, learning, sharing, supporting. And wonderful things happen. Here at Surrey, we never stand still.
Hello everyone. Welcome to the third event of the What It Takes series. I'm David Blackburn. I'm Professor of Virology and Head of the School of Biosciences and Medicine here at the University of Surrey. And today we're going to hear about what it takes to beat a pandemic. And I've got two very esteemed and experienced colleagues who are eminently able to explain indeed what it takes to beat a pandemic. Before I introduce my colleagues, let me explain what we're doing around COVID-19 research in the university. We have a number of colleagues working on the virus and on the consequences of the virus infection. And we call the virus SARS-CoV-2. That causes the COVID-19 disease. So COVID-19 is the name of the disease. We've got colleagues working on testing a vaccine, for example, in our clinical research facility in the school. That's work headed up by Professor Simon Skeen, working in collaboration with Imperial College London. So we're running a clinical trial on the effectiveness of the Imperial vaccine. Professor Margaret Raymond is working on selenium, and Professor Susan Lanham-New on vitamin D in the context of COVID-19. And Professor John McVeigh works with the Royal Free Hospital on developing a COVID-19 intervention, so a treatment for patients who are in the intensive therapy units. And other colleagues, Professors Nicholas Locker and Jill Elliott and Dr. Rachel Simmons are working on various other molecular, genetic and cell biology studies of the virus that causes COVID-19. I'd like to acknowledge the considerable efforts of all staff in the NHS during this very challenging, unprecedented time. But in particular, I'd like to share my thanks to those alumni of the University of Surrey, those healthcare workers who have graduated from the University of Surrey and are on the front line in this battle that we're undertaking against the pandemic. Okay, so let me introduce Professor Deborah Dunn-Walters and Dr Natalie Riddell. Professor Deborah Dunn-Walters is our Professor of Immunology in the School of Bioscience and Medicine, and she chairs the British Society for Immunology Task Force on Covid-19 and Immunology. And Dr Natalie Riddell is lecturer in Immunology and Ageing. These colleagues will share their knowledge on the important topic of vaccination. I'd like to remind you that any questions can be submitted through the question and answer function, the Q&A function, and we'll try our best to get through as many of those questions as possible. So, make yourselves comfortable, sit tight, and I'll hand over to Dr. Natalie Riddell, who's going to explain the importance and the mechanisms behind vaccines. Thank you.
Thank you, David, and welcome everyone. It's my pleasure to be talking to you this afternoon on COVID-19 vaccines. So during this very short presentation, I would like to discuss the types of vaccines that are being developed, the phases of clinical trials and how these prove the safety and effectiveness of vaccines, and also some analysis on how the vaccine priority order has been determined within the UK. So unsurprisingly, there's an array of vaccines that have been developed in response to SARS-CoV-2. I'm going to focus on the nucleic acid and the nucleic acid and viral vector-based vaccines today. And that's because the Oxford AstraZeneca vaccine is a viral vector vaccine and the Pfizer-BioNTech vaccine is a nucleic acid vaccine. And these are the two that have received emergency authorization to be used within the UK. So this is a really nice infographic on how viral vector vaccines work. It was produced by the British Society for Immunology. So the British Society for Immunology have been working really hard on trying to improve the way we communicate information associated to the pandemic and also to vaccines to the general public. So this is one of their infographics. And here you can see this is a SARS-CoV-2 virus particle. The green squiggly line is the genetic material of the virus, and it's inside. It's enclosed inside this black circle, which is the virus envelope. And on this envelope, you can see this green spike protein. And I'm sure most of you have heard of this green spike or this spike protein. It's the protein that allows the virus to gain entry into our cells and establish infection. So it's a very important protein. So the way these viral vector vaccines work is that the genetic code just for the spike protein is taken and it's incorporated into an unrelated, harmless virus. And this virus is used to deliver that genetic code, just the code for the spike protein, into our cells. So when we're vaccinated, this genetic code is delivered to our cells via this unrelated, harmless virus. And once inside, this code is able to result in the production of spike protein, which is then expressed on the surface of our cells, and that induces an immune response to that protein. So both B cells and T cells respond. The B cells are very important for producing antibodies, and antibodies are able to bind to free-floating viral particles and stop them from being able to gain entry into our cells. And the T cells are very important for orchestrating the immune response and helping B cells. And also they're able to recognize infected cells and destroy them. So both the B cells and T cells are really important in generating a robust and potent immune response. And very importantly, our B cells and T cells, they're very long-lived within humans. So they can last for years. And that means that they're able to provide protective immunological memory in case we ever become infected with the same virus again in the future. So in the case of the vaccine, we're producing an immune response to this spike protein. So in the future, if we become infected with SARS-CoV-2, these T cells and B cells directed against the spike protein will recognize the virus-induced expression of spike protein and be able to eliminate the virus before it can actually establish a strong infection and cause disease. Importantly, these B and T cells, it takes a while for their response to be generated. So it takes about 10 days to 2 weeks. So that means if you become infected with the virus, you have this lag period of 10 days or 2 weeks before you'll start to see this very potent part of the immune response. Whereas if you're vaccinated against the virus, previously, you already have these cells present within the person, and they'll be there able to quickly respond. So you won't have this lag period in the immune response to the virus, and therefore it will stop the vaccinated person from becoming ill with the virus and developing COVID-19 disease. So examples of the viral vector that are already in use is the Ebola vaccine, and now the AstraZeneca vaccine has been licensed for use in humans. And another important consideration about these vaccines is that, especially for the Oxford AstraZeneca vaccine, is that it can actually be stored at normal temperatures, so normal refrigeration temperatures, which is really important when we think about the logistics and how easy it's going to be to roll out a large vaccination program. So the other type of vaccine that's been generated in response to COVID-19 is a genetic vaccine. So this again uses the genetic material that codes just for the spike protein. But this time the genetic material is encapsulated or held within inside a fat bubble or a lipid bubble. And this fat bubble is able to merge with our cells and induce and therefore put the viral genetic material into our cells, where it can again then be made into the spike protein, which is then presented to our immune cells and again generates an immune response, a long-lasting immune response. So the only examples of genetic vaccines which have been licensed for use in humans are the 2 COVID-19 vaccines that have been produced by Pfizer and Moderna. And one of the things to consider about these vaccines is that because of it doesn't involve using a live attenuated version of a virus, you don't have to culture large amounts of virus. You just have to make this genetic material. So it's actually it's reasonably cheap to make and quite quick to develop. So because of because of how new this technology is, this mRNA technology is, there's been some concerns about the safety of these kind of vaccines. So I just wanted to show this table, or a couple of tables, which show all the clinical trials that use mRNA vaccines. This one is clinical trials using mRNA vaccines against infectious diseases. And this was up to and including May 2017. And this data was collected by the National Institute of Health in the US. And you can see that there's a large number of mRNA vaccines which have actually already been trialed within human populations by a number of different institutes and companies. And on the next slide, I just wanted to draw your attention to the fact that Medina has been developing our mRNA vaccines against different viral infections. On this slide, it shows a number of clinical trials using mRNA vaccines against cancers, and there's even more clinical trials have been done using mRNA vaccines in order to try and help fight cancers. And again, I've just zoomed in here so that you can see that BioNTech, the company that are working with Pfizer, have been using this type of technology to fight cancers for a number of years. So there's been a number of clinical trials already done using mRNA vaccines. So there's already a large amount of data on the effectiveness and also on the safety of this type of technology. So it isn't something that's brand new. Both the RNA vaccines and also the viral vector vaccines have actually been under development for decades or sometimes even longer. I think the first mRNA vaccine was trialed in animals back in the early 90s. Okay, so this just shows the different phases of clinical trials. So the definition of a clinical trial is the testing of a drug or a treatment intervention in a human population, in human participants. So you've got 4 phases. The first phase is really just checking the safety of the drug, or today we're talking about vaccines. So it's usually done in a very small number of individuals. So you can see around 10, 20 healthy individuals. And here they're really looking out for any unexpected side effects. So if you're giving someone a vaccine, you expect that they may have some pain at the site of the injection, that there might be some swelling. You're inducing an immune response, so they might even get a bit of a raised temperature or feel a little bit tired for a few days. But what they're really looking for is unexpected side effects or any serious adverse events. They often also check the amount of the vaccine that they can give. So they'd have like a dose escalation where they give slightly more to each individual until they reach the amount of the dose of the vaccine that they believe will be used in the actual final vaccination. So the second phase of clinical trials is where they check how well the vaccine actually works, so whether it is any good at preventing disease. So this is usually done in a couple of hundred people. And then as long as phase 1 and phase 2 have all gone well, you'll have phase 3. So this is confirming the findings, so confirming that the vaccine is actually safe and it does induce the desired effect, but this time in a much larger population of people, so usually well into the thousands. And if all this goes well, the results will be scrutinised and assessed by independent regulatory authorities who will then either authorise the vaccine to be used on the general population or not. But then the assessment of the vaccine doesn't stop there, it continues. So the safety of the vaccine as well as the effectiveness of the vaccine will carry on being monitored. So just to give you an idea of how these numbers compare to what's been done in the Pfizer and the Oxford clinical trials. So the phase 1 BioNTech clinical trial was performed on 60 individuals, and the Oxford trial, the data that they released in the Lancet at the end of last year, was looking at over 1,000 individuals. Half of them were actually vaccinated with the COVID vaccine, half with another type of vaccine. So these numbers are larger than what's generally accepted as being the standard amount for a phase 1 trial. And again, if we look at the Pfizer phase 1, 2 and 3 combined trial, for the phase 1 parts, they had 90 adults. For the phase 2, they had 360 adults. And for the phase 3, they had 38,000 adults. So these numbers are, well above what you could kind of normally expect. And the same for the Oxford phase 3 trial. There was 23,000, almost 24,000 individuals involved in this trial. So there was certainly no cutting of corners during the clinical assessment of these new vaccines. So how then was the development of these vaccines fast-tracked so much? Well, there was lots of players, and this is not in any particular order, but you can see here that, first of all, we had scientists, and that is that there's a lot of scientists have contributed to the work. They've switched their focus to tackling COVID-19 and SARS-CoV-2 infections. So they've been working round the clock, extra long hours. They've been working in collaboration. And also all of the other assessment authorities and the different agencies have been working rapidly with them as well. So this graphic over here is from the European Medical Agency and just gives you an example of how they fast-tracked their procedures. So the outside line is the normal process, and the inside line is the COVID-19 process, sped up process. So you can see usually a clinical phase will happen, and the researchers and developers will put together all the information, submit it to the agency. They'll then review it and they'll get back, and this takes around 210 days or so. Whereas in the current pandemic, the researchers are able to submit their results on a rolling cycle. So you've got every 14 days they're submitting data to the agency. So that means that when they get to the end of a clinical trial phase, there's the amount of time the agency needs to go through the data is greatly reduced. So they're getting a quicker response. So this is just an idea of how the whole process has been sped up. Another big issue is money. So usually the developers will have to complete whatever phase of the trial, and then they'll have to raise money to support the next phase of the trial. And this preparing a bid to get future funding can take up to a year or well over a year. And in between that time, all the research will just come to a standstill. In the case of SARS-CoV-2, money hasn't been any object. The governments have basically hedged their bets and put money into the development of vaccines in the hope that they will be successful. Another big factor is the environment, that you can't develop a vaccine and then test it until there is actually an epidemic in progress. And there hasn't been any problem with this because there has been such a large the virus has been spreading so widely in the general population. Luck is a big part of this. The S protein is actually a really good target for the immune system. It's very good at activating the immune response, which makes it a fantastic vaccination target. And finally, and by no means least, is there hasn't been any issue with recruiting volunteers to the trials. Tens of thousands of people have put themselves forward. And all these things have combined to helping speed up the process of vaccine development. So this is a really nice graph of the Pfizer data showing how well the Pfizer vaccine works. It is the Pfizer data, but it's been simplified and presented perhaps a bit more nicely by the New York Times. And what you can see, this is the number of weeks after the first dose, and this is the cumulative. We're looking at the cumulative incidence of COVID-19 disease amongst the clinical trial participants. So I just want to point out here, this is the number of people that actually became ill with COVID-19. So they had to become ill and then they had to test positive for the virus. So it wasn't picking up anyone who was asymptomatically infected. But looking at the development of disease, you can clearly see that these two lines diverge. So within the first, as I mentioned earlier, it takes 10 days to 2 weeks for the T cells and the B cells to start working and producing a potent immune response. So the first 10 days or so, both the placebo group and the COVID vaccine group, the incidence of infections track quite well. But from when the immune system really starts to kick in, you can see these two lines diverge. And not many more people go on to become infected in the vaccine group, whereas a number of people within the placebo arm of the trial continued, the number of infections continued to rise. And very similar findings were also found for the Oxford AstraZeneca vaccine. So here the red line is the COVID vaccine group and the blue line is an unrelated vaccine control group. And the two different graphs, the one on this side shows the incidence of infection, of disease, after 20 days after the second dose of the Oxford vaccine, whereas the graph on the right-hand side shows the cumulative proportion of participants that went on to develop these disease after the first dose. So you can see even after receiving one dose here that you still see a clear difference between the rate of infection between the two different groups. This as well as the ability of the immune response in the Pfizer vaccine to protect you following 10 days after the first dose. Both this information was used to help support the government's current guidelines on giving everyone their first dose of vaccination and then leaving a longer gap between the second dose in order to vaccinate more people quickly. And that's because of, you can see that you do get a good level of immune protection from the first dose. Okay, so I just wanted to also show you, this is a table looking at the priority of vaccination, the different groups and the priority that each of them have been given to receive their vaccination within the UK. So we've got 9 different groups and the table shows the number of deaths within each group due to COVID, and this was up until sometime in November because this analysis was released at the start of December. It also gives you the cumulative percentage of total COVID deaths attributed to that group. And then at the end here, this is what I really want to draw your attention to, is the vaccinations required to prevent 1 COVID death within that group. So what this means is that for every 20 vaccines given to people within the care home, you're able to save 1 life. For every for the over 80 age group, so the second priority group, you need to give 160 vaccines to save 1 life. And then for the over 75s, it's 350 people that need to be vaccinated to save 1 life. So using the data collected on the deaths and how many of them were associated to all of these different high priority groups, they were able to calculate where the vaccination would have its biggest impact. And this has really largely driven the decision of the priority order for the current vaccination programme within the UK. And then I come on to this slide, which I feel really grieved to have to show, but apparently it is a increasing issue, and that is that there's COVID-19 vaccine fraud. So I just wanted to point out that you should never be asked for payment. The vaccine is always free. You should never have to give out your bank details. No one should ever just arrive at your home to try and administer the vaccine, and you should never be asked to supply identity of who you are, such as a passport, by sending copies. So please just be aware of these nasty individuals that are trying to make money out of a terrible situation. And I'm just going back to this slide simply because I didn't want to finish on a negative. The vaccines have been developed unbelievably quickly and with amazing results, and it really should, the whole community, the scientific community, all the volunteers that have taken part, all the people that have invested in the vaccine should really be commended for how hard they've worked to get us to this point so quickly. So I just wanted to finish on a high note, and I will finish there. And now I shall pass over to Professor Deborah Dunn-Walters, who is going to facilitate the Q&A session, which I'm very much looking forward to. So thank you very much.
Thank you very much, Natalie, for doing all the heavy lifting there and putting all that information together in such a nice and clear way. And of course, I know you waited to the last minute to put all these things together because things move very quickly. And we've just this minute got breaking news that there's another vaccine that's announced their phase 3 results, which is Janssen, it's a similar vaccine to the Oxford vaccine that Natalie explained. And they've said that that's 66% effective in their preliminary phase 3 results. And late last night, Novavax released a press release. We don't have the data on this yet, so we much prefer to look at preprints and actual data on these things. But there's a fair amount of data indicated in the Novavax press release, and they say that 89% efficacy for their vaccine, and that's a different one again, which is a protein vaccine with a novel adjuvant. So it does seem to be coming quite thick and fast now, the different vaccines that are going to be available to us. And I should say that the UK have pre-ordered 60 million doses of Novavax and 30 million doses of Janssen. So our options are becoming more diverse. We've got a lot of questions. We had a lot previously, and I've noticed that there's quite a few coming through now. So I'm not sure whether we're going to have time to go through them all. I've tried to do a bit of grouping, but we're going to do our best. I'm going to start first of all. If I ask you, Natalie, there's a question from John and Sarah that says, after vaccination, what is the best way to find out whether you've actually acquired immunity? Because all the vaccine manufacturers state that not all those vaccinated will be immune. Is there a valid test?
Well, the short answer is, in terms of a valid test that would be available on the scale that you'd need to test everyone who'd been vaccinated, there isn't such a test that we can do on that level at the moment. The best way to look for protective immunity is to see whether neutralizing antibodies have been produced. So these are antibodies that not only bind to the virus, but they actually block the virus from being able to gain entry into our cells and establish infection. But to do this in the laboratory is actually quite a time-consuming and costly task. So you just can't do it on the level that you'd need to check everyone that's vaccinated. Any other comments, Deborah?
I think that's it on that one. The government has put some money towards encouraging people to try and develop some tests that might be scalable, but these things are not easy in a short time frame. So at the moment, I'm afraid we don't know on that one.
The best thing really is to keep up the social distancing, keep wearing face masks, keep washing hands, following all the guidelines until we know that we're in a situation where, thankfully, we don't have to do that anymore.
That's true, yeah. So I've got quite a few questions in advance, which I've grouped because they're all on a similar topic. And I kind of titled the topic, Protection from Disease versus Protection from Transmission. So I'm going to have a go at explaining that, because there's still quite a lot of confusion around. When we try and communicate science to the public, we sometimes forget that everybody, we've got people who don't do O-level biology, they do physics instead. And so these things can be quite, sometimes we don't explain them very well, basically. And the thing is that immunity is not all or nothing. And this comes through when the companies are talking about we've got 90% protection here. That means that only, that 9 out of 10 people will be protected and 1 won't. And it depends, of course, how you measure these things. But within a person, there will be degrees of immunity. And if you get like 100% effective immunity that will stop the virus coming into the body, or, it's given very short shrift and not allowed in for any length of time at all, and then you won't get disease. But obviously, if you encounter the virus somewhere, you do get it and your immune system has to stop it. And how long your immune system takes to stop it will depend whether you go on to have the disease or not, or whether perhaps you don't have the disease, but you still have it. So it is possible for you to be protected against disease because your immune system's there to come in and fight it and stop it. But for a time, you would walk around still having the virus in your nose and mouth. And I think this is illustrated by the fact that people have measured virus in people's noses for 3 days before they actually get symptoms of the disease. So if you could imagine that kind of scenario, only if you've got protection from a vaccine, you might not even get the disease. You might turn into an asymptomatic carrier. And so that's what we're meaning when we say that there's a difference between protection from disease and protection from transmission. So this has led to other questions from other people about, will the vaccine prevent me from infecting other people? I don't actually have a name on that question. And depending on everybody's immune systems, it's not going to be 100%. We can't say that the vaccine is going to prevent you from spreading the disease. It will have an effect on transmission because it will in some cases. But at the moment, I think the AstraZeneca, they did a really wide range of different tests in their phase 3 data, which meant that the data was quite confusing to try and pick through. But one of the things they were looking at was looking for asymptomatic infection. And I think they said there was perhaps 50% efficiency at stopping you carrying the virus. But they were just measuring everybody once a week with a nasal swab for the PCR test. Some other data last week, I think it was, out of Israel was quite encouraging, that at day 21 after the Pfizer vaccine, they found a 60% drop in the number of cases of infection. So, people were saying, oh, it's not as good as Pfizer said it was, because, the numbers for efficacy of Pfizer have been touted around at 89% and 95%. But that's measuring protection from disease. That's measuring whether you get symptoms of the disease or not. And someone else had the question, why do people measure that? And quite simply, when you're trying to run a clinical trial with tens of thousands of people, it is hugely expensive. I mean, this is millions and millions and millions of pounds anyway. And it's an easy endpoint to say, did you get symptoms or not? The endpoint, how bad were your symptoms, is more difficult to measure. The endpoint, did you have the virus, is more difficult to measure. So all these add a lot of extra work into what is already a big and busy clinical trial. So the primary endpoints for a lot of these things were, did you get disease or not? And all these other questions come in afterwards. So I think that was a question from Bashir. There was a question from Jan, which I think I answered, talking about 46% efficacy in protection from transmission, and also the new data from last week from the kind of real world data in Israel. Sue says, when do you think we'll know for sure whether vaccines will prevent people from transmitting the virus or not? We are still waiting on data coming in. It's still quite early days. You know, we only started vaccinating people kind of mid-late December. And so, you know, and for the first 2 weeks, nothing happens anyway. So it's still quite early days in this, and everybody is scrambling to collect whatever data they can to answer these kinds of questions. But we do know that, you know, if you've got 95% protection against disease, the chances are you're not going to have 95% protection against carrying the virus, which means you could be personally protected and feel a bit relieved for that either you won't get the disease, or if you do get it, it'll be milder. But that doesn't mean that, you know, you can go around visiting 30 different houses, because if someone in the supermarket had infected you, you would then carry it around those 30 different houses. So this is why the government's really keen to say, please don't change your behaviours just because you're vaccinated right now. Also, I guess, because it's really important to note that first 2 weeks, there's no protection at all. On Natalie's graph, you could see there the first, you know, 10 to 14 days, the vaccinated people and the placebo group, the lines were on top of each other. It takes a while for the immune system to get up and running. Bashir also said, what new lessons are emerging from real life applications? So I think I answered that about the Israeli studies. It is being monitored very closely. I have to confess, because there's also a question here about what about the other vaccines we hear very little of, Russia, China, India. I have to confess, if our government hasn't pre-bought some of these vaccines, I haven't been tracking them to look at their progress, because as it is, it's quite difficult to keep up with everything that goes through. So I'm afraid I don't have information on those. So I'm going to take a break and ask Natalie now to answer the question from John. Once you've contracted COVID and you've recovered, why don't the antibodies that your body produce protect you from contracting the infection again?
Okay, yeah, good question. So the thing is that not everyone produces the same level of antibodies, and not all antibodies function the same. So this means that where some antibodies are able to bind to the virus and stop them from being able to gain access into our cells, other ones aren't as good at doing that. So yeah, it's hard to predict who's going to have generate antibodies which are going to protect them from future infections, and people that perhaps aren't going to generate such a good antibody response. Yeah, did you have anything you wanted to add to that, Deborah?
Yeah, to be honest, the correlates of protection that we measure, we can measure things in our blood in immunology and say, okay, we think it's neutralizing antibodies that are the thing that you measure in order to check whether you've got immunity. It goes back to that other question about how difficult things are to measure in a real life scenario. And there could be all sorts of other things that we can't measure at scale, or we haven't looked at yet, that could also be involved in some of these things as well. So I think that's what's keeping a lot of immunologists busy.
Yes, the other point is that we don't know at the moment exactly how long these, if there are protective antibodies, how long they last for. There's data coming out, and I think you sent something through yesterday that said they could see protective antibodies for up to 30 weeks, which is showing that they don't disappear after the first couple of weeks. But the data isn't there yet to kind of say how long any immune protection might last from natural infection, or indeed from the vaccine. So this is where work's still going on to figure this out. I think that possibly also plays a role in, someone mentioned, I remember seeing a question about vaccine passports and how they might work. And I think at the moment is we actually don't have enough information to be able to produce vaccine passports, because even if someone has had a vaccine, we don't know whether they're able to, they could still transmit the virus, as you've just given us a really good kind of description of. And also, even if the vaccine does induce a immune response which stops people from transmitting virus so well, we don't know how long that immune response is going to last. So how much value you'd get from a vaccine passport at the moment is it's just not known.
It brought me to another question someone asked about presence of virus in the population, and should should we just be looking at our values when we're deciding, whether we can lift restrictions and things like that? And why can't you get back to normal just because, can the people who've been vaccinated go back to normal? And we're in a situation when there's so much virus around in the population that we've got to really take care, because there are all these unknowns that we don't know how you'll be able to pass it on. It's a question of balancing risks. And one of the biggest risks is the fact that there's just so much of it out there. And while we're still in this kind of ramping up on getting everybody vaccinated situation, we have to make sure that there's not lots of virus replicating away in the population at the same time as we're vaccinating. And I think that goes to questions about viral variants as well that have been coming in. And maybe I should tackle the thorny issue of the delay that the government announced between the first and second doses. One of the questions that was raised by quite a few people is by delaying the second dose, so you don't have as good a protection for the 12 weeks until you get the second dose, at a population level, are we encouraging viruses to come up with new variants because we haven't got the best immunity? And because there were so many questions about that, a lot of people looked very carefully. And one of the main reasons for viral variants arising, people think, is just that it is in the population, and there's so much of it around, that there's so much viral reproduction that it's got more chance of coming up with variants. The variants that arose before the vaccines were rolled out. That's not to say they weren't out there, because there were all these people taking part in trials, but I know of nothing that's linked the variants with vaccination or partial vaccination or anything. So the main thing we need to do, vaccines are going to save us, but it's not the only thing. We've still got to stick with this lockdown for as long as we can on that. There's another question, Natalie. I don't know if you've got anything on what information is available and where on which vaccines are not suitable for people with hen's egg allergy. I don't know if you want to comment on the kind of the composition of the current vaccines.
So I mean, the best people to ask before, if you're worried about having a vaccine and having an allergic reaction, the best thing to do is to actually raise it with your GP, because they will know the ingredients that are in the vaccines and they'll be able to advise you on it. But we can also, we know that the Pfizer vaccine and the AstraZeneca vaccine didn't use eggs in the production. So as far as I'm aware, they should be safe for someone with an egg allergy. But as I said, I'd definitely check with your GP. Yeah. I don't know so much about the protein-based one, the Novavax that's just been reduced. But yeah, for the Pfizer and AstraZeneca, they weren't made with using eggs.
Yeah. There's something called the Green Book, which the government and produces, the NHS, the Department of Health and Social Security, which is like the Americans called it the playbook for UK doctors. It's got a whole chapter on COVID-19 and what it is and how to do the vaccination and everything else. And so the information from the MHRA and JCVI and all these regulatory bodies about how to give the virus and what to look out for, they update the Green Book all the time, and the GPs refer to that. And so your GP best knows your medical history and then can look at the Green Book advice in light of that. Here's one for you, Natalie. On the basis that we're likely never to rid ourselves of this disease, can you please let me know if we're likely to need a vaccination each and every year going forward?
I would guess that that's a highly realistic possibility, similar to how we have a seasonal flu vaccine, that we might need to have something similar for COVID, to protect us from COVID. So we don't know yet. We can't say for definite, but yes, it's highly possible.
And in my list of questions, I've come to a section now on delaying the second dose, because I know there's a lot of people have questions on this. it did come as a surprise to me, I have to say, that that the Pfizer vaccine was included in the change of policy, changing it from a 3-week interval to a 12-week interval. I'd kind of taken it for granted that it just applied to the AstraZeneca one, because I know they have data that, if anything, there may be an improvement in the result by delaying that second dose by 12 weeks. So for the AstraZeneca, we could see that there was data to support that. And to find out that they decided to do it with the Pfizer as well was quite a shock initially, because as scientists, we like to follow the data. But then, you know, the JCVI explained that there's a shortage of vaccines to roll out quite quickly. And I have since seen modeling that they did. So there's a lot of modelers employed on COVID-19 situations in epidemiology and everything else this year, to actually say, how do we save the most lives? And so you kind of put in the model what the vaccine efficiency is if you just have one dose, and vaccine efficiency if you have 2 doses. And then they've modeled what that effect has on saving people's lives, if there's a crossover at any point, whether the policy might change. But of course, this is all dependent on the real life data coming out. The CMO's office have put in place with Public Health England and the Test and Trace a linked system now. So the pillar 1 and 2 testing is now linked to the vaccine data. And so they are collecting data in real time on how effective the vaccines are. But of course, you know, if the Pfizer vaccine, was rolled out before Christmas, like I say, for the first 2 weeks, nothing happens. And then after that, you then start looking at the data to see if there's any difference. So it's quite new. We wouldn't expect any information on that for a couple of weeks, and maybe a couple of weeks after that for the AstraZeneca. But they are collecting the data and looking at it, and have assured us, because we've had conversations with the BSI and the CMO's office, that if there were any change in anything, then they would change the policy. So if it looked like this was a really bad idea, they would change the policy. There was a small amount of data that you could look at in the kind of weeks 2 to 3 data for Pfizer, which said that it was 89% effective at that point, and it's 95% effective after 2 doses. So you effectively, you know, if someone gave me just 200 doses and said, okay, go into your local hospital, you can choose to protect 95 of these health workers or 178 of them. That was basically the choice. But they are looking at it very carefully. Also, there was a certain amount of, you know, just drawing on experience from immunology and from previous vaccines, that quite often a vaccine would have a longer period between the first and second dose. And I don't know exactly what was going through the minds of everybody when they decided their protocols for phase 3, but I'm pretty sure that how fast can we get these vaccines out was a factor in there. And so they were choosing the smallest possible intervals that they could in order to do the trials and get the information out. With this other data, which admittedly is from people who've been infected, that Natalie just referred to, you could see that after the initial big drop in antibodies, because in a disease there's a huge amount increase in antibodies and then a big drop again afterwards. Once that's calmed down, you find that there's some really good antibodies underneath which are persisting. The half-life of that is 30 weeks. So it shouldn't be a problem waiting 12 weeks for the second dose. So people who asked questions on that, there was Helen asked a question on that, Bashir, Anne asked a question on that. I think I've answered everything that they asked. Oh, there was one answer about there are controlled trials. So Anne asked if there were going to be any trials carried out. So the CMO was adamant that he wasn't going to to do randomized controlled trials on old people when we already knew that vaccines were effective and that they were at such high risk. But presumably on younger groups of people, there is a plan to do controlled trials looking at the difference in the interval between 2 doses, but also, and this goes to another question that came through today from David and Phil, about what we call heterologous vaccination, where you do your first dose with one type of vaccine and your second dose with another type of vaccine. And at the moment, that's only advised in this country if it's an absolute emergency, if someone needs a second dose and they don't know what the first dose was. But if I was setting up an experiment to see if I could improve efficacy of a vaccine, that's exactly what I would try to do, is to see if I could prime with one and boost with the other, and that they would synergize together and get an even better result. And there's kind of some good hypotheses about why that would work. And so they're going to actually be trialing that as well. So Natalie, do you want to, this is a bit speculative, I guess. There's a question from Peter, which says, what's been learnt about the development of the COVID vaccine? Can we see the development of a whole range of new vaccines, some of which would cure people rather than using antibiotics? I guess maybe talking about bacterial vaccines there.
Yeah, I guess that's possibly one of the good things that's come out of the whole COVID pandemic, is that, it really has moved science forward and vaccination technology forward as well. So the potential to use the mRNA vaccine technology for other infections, including bacterial infections. It's definitely a likely possibility that we can use the same technology for a wide range of infections. So in terms of, I guess, antibiotics, you use those as a treatment once someone is actually infected. So vaccines are all about preventing someone from becoming infected generally, apart from perhaps in the case of cancer vaccines. But I'm sure there'll be a lot of work, well, there already is work, but there'll be a lot of work looking at how this new technology can be used against other infections, in order to, hopefully, if we can prevent people from catching bacterial infections, then we can bring down the amount of antibiotic use, which obviously is a very desirable thing to do, as we're seeing so much antibiotic resistance building up within different microorganisms.
Yeah. So you mentioned cancer vaccines there, and it was in your talk as well. And there's a question somewhere, I know I've seen it, I can't exactly find it, but someone was saying, could you just give a little bit more information about what a cancer vaccine is?
Yeah, so a cancer vaccine, these are actually vaccines to treat cancers as opposed to prevent cancers. So it's not kind of giving someone a part of a cancer, like a cancer protein, thinking that they may develop that cancer in the future. But this is more, our immune system should protect us from cancers. It should recognize, because of their cancers are mutated cells, we should recognize, the immune system should recognize cancers as foreign and should develop an immune response to them. However, obviously that isn't the case, because we see such high prevalence of cancers, and that's because of often the cancers are very similar to self cells, so our immune system kind of tolerates the mutated cells, or it can be that the cancer produces an immunosuppressive environment which stops our immune system from responding. So in this instance, there's a lot of work going into developing vaccines which are actually kind of cause an immune response to the cancer in individuals that already have cancers. Hopefully that's explained a bit of the difference between it. They're not preventative, they're more treatment in individuals.
Yeah, thanks. We've got a question here about from John, who says, is antibody-dependent enhancement potential already proven to be negligible for each of the vaccines? And if not yet proven, what are the possible associated risks and implications? So I think here I probably have to explain what antibody-dependent enhancement is. And for some diseases, it's been shown that instead of protecting against disease, the fact that you have antibodies against a virus helps the virus get into a cell. So, for example, if you've got a macrophage which has receptors for antibodies on the surface, so that it can these antibodies help the macrophage recognize the virus target. But if the virus lives in a macrophage, then it's kind of helping the virus to get into the cell. And so antibody-dependent enhancement is if you've got antibodies already to the disease, then you can actually cause a worse disease second time round. And of course, everybody was really worried about that. I mean, this happens with dengue sometimes, which is why it's been very difficult to make dengue vaccines. So of course, there were a lot of people looking at this. And in the early days, people were looking at all the different types of antibodies that were being made in patients that had COVID, and they were checking to look for antibody-dependent enhancement. They're complex assays that are done in the lab, so they take a bit of time to do. So by the time it got to about June last year, there was a huge amount of data which you could review, and nobody had found any evidence for that kind of antibody-dependent enhancement where you actually enhanced infection of cells by having the antibodies around. There's a kind of another definition of antibody-dependent enhancement could be that by having a lot of antibodies around, these are helping stimulate other cells of the immune system, which are producing cytokines and contributing to what some people have referred to as cytokine storm. We know this is the pathology of this disease is highly inflammatory. So there may still be aspects of that during the disease itself, when the immune system has got so overexcited because it couldn't get rid of the virus first time round. It's got a massive amount of inflammation going on. It's a hypothesis that there might be that kind of pathology going on. But as far as a vaccine goes, we've got no evidence of antibody-dependent enhancement. That's not to say that people are going to stop checking, because we're going to get next generation vaccines coming out very soon. I mean, I think Novavax last night in their press release already said that they're working on the next generation to go for one of the other variants, as have, I think, Moderna in the last few days said that as well. So people are going to be constantly monitoring. It's one of the things that they're really concerned about, just to make sure that that's not a concern.
I guess in the data we have so far, even though it's only limited, just to add on to that, they found that the severity of disease, as much as people in the placebo group have contracted the virus and developed symptoms more than people in the COVID vaccine groups, but also the severity that people in the receiving the vaccine haven't had severe disease. And if you thought you was enhancing it, I know, like I said, it's in very small numbers, but there hasn't been any indications from the data thus far that there's any enhancement in disease if they do become ill.
Yeah, no, that's a really important point. And yeah, thanks for that, Natalie, because it's just me to say again that even if you do get the disease, if you've been vaccinated, the chances are it's going to be a lot milder than it would have been had you not been vaccinated. So that's a really important thing. We talk about, all these press releases are going, oh, I'm 66% efficient and I'm 98% efficient, etc. But the Oxford AstraZeneca vaccine doesn't have as high a percentage number as some of the others, but not a single person went into hospital in that trial, in the phase 3 data. And that's the important thing, they're measuring as an endpoint symptoms of the disease. And the symptoms could be loss of smell and a bit of a cough, and that's the end of it, as opposed to, severe disease where you need oxygen. And so these things are important to consider as well. And they were a much more complicated endpoint to measure in a lot of the trials. So we're still looking at the real world data about who's not going into hospital. I think the data coming out of Israel is saying quite clearly that it's having a big effect there. I don't know if you want to tackle pathogenic priming, Natalie. It was kind of a new phrase to me, but I think it's where the virus protein looks like a human protein, so the response is auto-antibody.
Yeah, I guess that's sort of what we call molecular mimicry. So where the pathogen is so similar in structure to some self tissue that if you get infected with whatever, like the virus, the bacteria, then the immune response directed towards that won't be able to differentiate between that pathogen and between our self cells. So you end up with kind of an autoimmune response. And there has been some evidence in SARS-CoV-2 there are potentially auto antibodies produced, but that's in the actual infection. In the vaccine, you're not giving the actual virus, I think, is made up of more than 20 odd proteins. The vaccines are just generating antibodies to one protein, the spike protein. So by having the vaccine, you're actually limiting the possibility of generating like all these extra antibodies and I guess reducing the risk of producing autoantibodies. So it's another reason to get vaccinated rather than, think that you'll gain immunity just from natural infection.
Yeah, I don't think anybody's seen any examples of molecular mimicry in the vaccine trials at all, but there's at least half a dozen papers now coming out and more people looking at autoantibodies in the disease. And in long COVID, I think yesterday or the day before, there was something on the preprint server looking at autoantibodies in long COVID. So again, it's early days. There's still lots of people working on getting the data on that. But for the vaccine, there's been absolutely no instance of that because it's just to the spike.
I think there was a question where someone asked about if you suffered from chronic fatigue or something along those lines, whether having the vaccine is feasible. I mean, kind of based on the discussions we've just had, and again, it's something you'd want to check with your GP and your clinical team. Definitely, we're not clinicians, we're scientists, so always take these kind of medical questions to your your doctors. But having the vaccine is less likely to induce these autoantibodies and things that may kind of have long-term effects on you compared to actually catching the natural infection. So even in people that are immunocompromised or people that have other conditions, it's still advised that you get the vaccinations rather than run the risk of having the natural infection, which is more likely to induce all these extra effects over and above the actual what having the infection causes.
Yeah, chronic fatigue syndrome. Kind of a difficult thing. You definitely need to speak to your own GP about that. Immunodeficiency is, there's lots of different types of immunodeficiencies, and the British Society for Immunology have actually put out a statement on immunodeficiency. The stock answer is, if in doubt, you need to speak with your GP because they'll have your medical records and know about you better than anybody else. But even if you think your immune system isn't going to make a good response because you're immunodeficient, it might still make some response, and that might still be useful. It's this kind of, it's not always 100% or nothing. You could have a small percent which could be useful to you as well. There's a question from, sorry, I should have said that, one of those questions was from Nicholas on the ME chronic fatigue, and the other question on pathogenic priming was from Rosie. There's a question from Jennifer, which says, is there any connection between air pollution rates and the current pandemic? Now, I know at Surrey, we've got some people working on air pollution, and I know, if you have a quick look in the literature, you can see that there are a few papers. I am a little bit wary of them at the moment. Again, maybe it's one of these things that's early days, because if you show a correlation between levels of air pollution in an area and levels of COVID-19 infection, you normally get higher levels of pollution in an area where you've got a lot more people. And when you've got a lot more people together, you get higher levels of infection. So, the correlation is not causation warning on some of that data, I think. There's one on the particular one, which we may have already covered, about can I play with my grandchildren once I'm fully immunized? 3 weeks after second jab. So we've covered a bit of this, that, we can't measure everybody's immune system to know that if you're fully protected from carrying the virus around. You have your own risks. And if you've got no vaccine, you've got a personal risk of being ill. And even the best vaccine at the moment was Pfizer, was 95% in phase 3 trials. I think Israel's just come out after vaccinating millions of people and saying it's 92%, which is good confirmation. But that's still, 1 in 20 people could get the disease. So that would be, your risk there, a personal risk. And then there's the risk to your friends and family if, we're in the situation we are with so much virus around in the population that passing it, ending up being an asymptomatic carrier is not really what you want to be. And then there's, in my mind, when I was thinking about this, there's also a wider risk in society that if we start getting like a 2-part society, that some people can go out to the shops and the gym and the hairdressers and others can't, that how on earth do you organize that in a society? So I think at the moment, it's going to be a question of when the R is low, so that it's going down, and the incidence in the population is low, then the kind of risks for the population as a whole can be assessed about coming out of the lockdown. But until then, I think we all have to, as Polly Toynbee put it, behave ourselves. So there's
I've seen 2 quite important questions, I think, on the live chat questions that are coming through. One of them is on the...
Oh gosh, there's loads.
So this was an anonymous question. The possibility that RNA from the vaccine could insert itself into the human genome, and this is a very quick one. It can't. RNA is actually very fragile, and it's taken up within our cells, and it won't last very long. Long enough to produce the protein for our immune system to respond to, but the RNA will actually be degraded very quickly. So that definitely is not an issue with the RNA vaccines. And another one I thought would be, as we're running out of time, Liz asked, could you please offer any comments on the fears that some people have about the vaccines affecting fertility, and whether pregnant women or women who try for a baby should get vaccinated? And one of our colleagues who works at Imperial College London actually put together the most up-to-date information on vaccines and fertility. So I'll just read out the overarching statement on this, and that is that the current advice is that people who are trying to become pregnant and those who are breastfeeding should receive the vaccine. I mean, if you're breastfeeding, there's a possibility that the antibodies you produce will actually go through the breast milk and offer your baby some level of protection as well. And also people that are pregnant should be offered the vaccine if they are at high risk of catching the virus, because there could be serious complications if they do catch the virus. So, for example, frontline workers that are pregnant could be offered the vaccine. And this is based on data. All this is based on data. There was people who took part in the clinical trials, and they were asked, the ladies of childbirth age were asked whether they were pregnant or planning to become pregnant. And they weren't to take part if they were planning to, but obviously some people then did go on to become pregnant, and there was no real effect on those individuals that received the COVID vaccine compared to the placebo. And there's also more animal data about receiving the vaccine whilst the animals were pregnant, showing that there was no real effect. So we are getting the data now coming through. So the guidelines have been updated. It's fine to get the vaccine if you're planning to become pregnant or breastfeeding. And it is probably advisable to get the vaccine if you are pregnant and are at high risk of catching the infection. But at the same time, this is a very personal decision. And again, it's something that you should be able to talk to your GP with and gain advice if you are worried.
So Julie says she's interested in the cause of higher death rates in males. Is it due to a difference in immune response and what's being done to improve outcome? Is it related to estrogen, and how does this relate to menopausal women? So if estrogen is known to affect some aspects of the immune system, but also the X chromosome is packed full of immune-related genes as well. It's not all just about positive effects of estrogen, and there also there may be some negative effects of androgen, of testosterone. And also the receptor that the virus goes for, ACE2, has actually been shown to be different between males and females in some instances as well. So it's quite a complex situation, but it is very clear in the data that men don't do as well as women. There was one question from John saying, can you give a guesstimate as to the chances of further pandemics this century? And I would hate to give a guesstimate as to the chances of further pandemics this century. I think what I would say is that a pandemic is an outbreak that escapes control, and that governments around the world all join in pandemic preparedness. And maybe if we're looking in the wrong direction, we're unlucky to be, concentrating on flu instead of SARS or something like that. Or if maybe we've got a bit complacent because the last time it happened was in 1918 or so, then, maybe this is where we've got to learn a lot of lessons, and there will be a lot of wash up after this. I think the government said last July that there was going to be an inquiry. But just as a warning, so Ebola, for example, was an outbreak that could have got out of control and didn't. But I mean, the West Africa had 28,000 cases, and then in the DRC there were another 3,500 cases just recently. 2017, Marburg disease, that was an outbreak. Lassa fever, that's still going on. It doesn't appear to be as infectious as would cause a problem. 2012, MERS, 2003, SARS, 2018, Nipah virus. So this is just a list of things that I just pulled off. These are outbreaks that have occurred, in recent memory, and because people have been on top of them, they've stayed as just outbreaks and not pandemics. So I think the lessons there is not to take our eye off the ball.
I'm just looking at some of the ones that are perhaps very quick to answer. There's one on how do the stats being quoted, i.e. the 69% efficacy, 90% efficacy compared to standard well-known vaccines. And as me and Deborah have both mentioned, the spike protein is actually really good at inducing an immune response. So actually like 65, 69% effectiveness is really good for a vaccine. So the flu vaccine, we all know is not so good at inducing a response in older individuals, about 30, 40% efficacy. But actually, even in younger individuals, it's only around 70% effective. So even at the lower end of the COVID vaccine effectiveness, they're generally better than some of the vaccines we have for flu, for example.
They're very good. If messenger RNA is cheaper and quicker to produce, why are the vaccines more expensive, the Pfizer one's more expensive than the Oxford one? There is the cold storage issue for both of them. But I'm not sure people know this about the Oxford vaccine, but none of the scientists involved in inventing that vaccine are taking any royalties from it, and the company is not making any profit from it. It's cost a fortune to produce, so they have to kind of recoup that, but they're not doing it on a profit-making basis. That's at cost. So that $3 a shot or whatever it is, that is how much it costs to produce the vaccine, and they've given a commitment for X amount of time that they're only ever going to produce that one at cost, because there's a lot of countries in the world that can't afford $19 a shot for some of these vaccines. So that's why a lot of companies are doing it on a business footing, but Oxford and AstraZeneca are not, and that's the main reason why there's a difference in cost there. Future strains, actually some data came through. I was reading this morning, so that's probably on the preprint servers. In fact, all week I've been reading things about effectiveness of vaccines and against the different strains. It's beginning to appear, both in the lab tests and in some kind of real life situations, that the UK variant of the vaccine, the one that was more transmissible and caused us so much problems, the existing vaccines work against that variant. In fact, the existing vaccines, I read somewhere that not only do they produce antibodies against SARS-CoV-2, but you can get a response against SARS-CoV-1. So, we never knew how much overlap there was because there weren't that many people got SARS-CoV-1, but there is some reactivity against SARS-CoV-1 with some of these vaccines. The variant that was more concerning was the one coming out of South Africa, and there's a similar one from Brazil. And the reason that was concerning was because the mutation that occurs in the spike protein for that one, the E484K mutation, is at the receptor binding domain, and they think that changes the protein shape in such a way that it's going to be able to escape. And in the lab tests, it does escape from some of the monoclonal antibodies. So monoclonal antibody binds in one place on the spike. So if that one place mutates, it's easy to escape from that, right? Convalescent plasma is like a library of antibodies that a person has made after they've had disease or a vaccine. And so that's a whole library of antibodies. They're still fixed, they're not going to change, but there's a lot of diversity in it. So can a virus escape from that? It's less likely. And there's information in the labs to say that it is less likely that a virus can escape from something like convalescent plasma, where you've got a whole library of antibodies against it. But it is still, it does escape from some of them. So if you've got a library this big, the South African variant, maybe only that many of them are going to actually be able to work anymore. So there is a reduction, it seems, for the South African variant. And looking at it another way as well, that when we're making an immune response, when another virus comes in that's a variant, our library that we make of vaccines and cells to the spike protein is not fixed. All these cells can jump off the shelf and go into another immune reaction again, second time round, and can kind of change themselves and alter themselves. So our immune response is not only diverse, but it's dynamic as well and can react in real time. So there are concerns that a couple of the variants, aren't going to be recognized as well by current vaccines. And this is why some of the people are already working on their next gen vaccines. So that was a question from Jana. Cancer vaccines. Oh, we've got about 1 minute left. Did you have anything, Natalie, that was?
I see there's some questions on nutrition or things that you can do to help protect yourself against infections. And I mean, basically, I think in a very quick summary, there's a lot of research going into different things that we can do, and the way nutrition may help improve our immune response to the virus. And there isn't really any, we don't know yet. The research is ongoing. However, to produce an immune response, it's an energy using process. It involves the production of many products. It involves our cells dividing, which means that we need to make lots of cell structures, and all of this requires energy, and also nutrients and minerals are often involved in the processes as well. They're needed for these processes to occur. So, I mean, taking a multi-nutrient is definitely, it's not going to harm anyone, and it could also be beneficial. So there's no reason why people shouldn't get a multivitamin tablet which covers a lot of, a wide range of vitamins, one of these A to Z type ones. At the same time, you definitely shouldn't take too much, because if you have too much of certain vitamins, they can actually be detrimental to immune function. So definitely don't go over the guidelines thinking that you might end up with an extra benefit. But I would suggest that there's no harm in taking a multi-nutrient.
So I'm just going to rattle through a few of these things. Someone says, my daughter had the Pfizer vaccine and tested positive 8 days later, but didn't have any symptoms. Could the vaccine have caused the positive test? It's more likely that, I mean, eight days after the vaccine, she wouldn't have been protected. The vaccine doesn't cause a positive test. Getting the virus causes a positive test. So it's more likely that she had an asymptomatic infection there. Are vaccinations effective for people with suppressed weak immune systems? We do think they will have an effect. We're just not sure how much. So yes, get vaccinated, but also do still keep taking your additional precautions. My son tested positive, then went to donate plasma, was told he didn't have enough immunity to be useful. Oh dear. But does it mean he's more likely to contract COVID again? I guess in a nutshell, yes, if you don't have as many antibodies, we think that you're more likely to get it again, but it would be milder if you did. It is rare, but people, they've looked at health workers and people do get reinfected.
We had one question which might be difficult to answer. It was an anonymous one saying, what advice would you give to someone who was still sceptical on taking a vaccine? And I guess it's based on where the skepticism comes from. But I mean, what we can say is that, and I hope this come through from the presentation, that these vaccines, although they've been put together quickly, there hasn't been any corners cut in terms of checking the safety of these vaccines. And so there really isn't safety concerns. We know that people that receive these vaccines, there's no kind of increased risk of serious adverse events in these individuals. And we know that it's able to reduce the amount of symptomatic infections in these people, and also that it reduces the severity of infections in people that do become ill with COVID. So, I mean, I guess it's where your skepticism is coming from, but I can't see any reason why people wouldn't take the vaccine, any of the vaccines that are being offered.
And from everything that we've seen, I think, taking the vaccine is a much better proposition than getting the disease and all that entails. One very, very quick question here, because I'm told we're out of time, is will vaccine apartheid between richer and poorer countries lead to deadlier variants, and what are the implications for current vaccines? So that would be quite a speculative answer, but I think it brings up an important point that maybe we should all take away and think about, that, in a global society, we have to also look to other countries that haven't got the facilities that we have for vaccinating their populations and help them, because it's a global problem and not a national problem. And it is something that we should take away and think about. But I'm afraid, sorry, Natalie, if you'd spotted any other questions that you wanted to tackle, but we really have reached the end of the line on time here. I want to thank everybody so much. So many people have joined in for this, for coming and for listening and for all your questions. And I'm sorry we couldn't get to them all. There were just so many of them. I want to thank Lucy and Anna and Sam from behind the scenes, from doing all the organisation from this. Everybody else for coming. Natalie, for your brilliant presentation. And till next time, thank you. Goodbye.